My name is Ben Sawyer, and I'm going to be presenting on chemoreception in terms of taste and smell. Olfaction is a form of chemoreception, which is the ability to perceive specific chemical stimuli, and in this case relates to smell. Similar to somatic senses, like the perception of touch, vibration, or pressure, chemoreception relies on receptors to transform information about the environment into action potentials that can then be relayed to and interpreted by the brain. The olfactory bulb, which is part of the forebrain, receives input from primary olfactory neurons in the epithelium in our nose and synapses on the bulb to decipher what we smell. Our olfactory system contains epithelium embedded with olfactory sensory neurons, whose axons form cranial nerve 1, also known as the olfactory nerve. Cranial nerve 1 synapses with secondary neurons in the olfactory bulb in the frontal lobe. Neurons project from the bulb through a tract to form the olfactory cortex, which bypasses the thalamus. Processing of smell occurs in the olfactory epithelium, as well as the olfactory bulb and other areas of the brain. Ascending pathways from the bulb lead to parts of the limbic system concerned with emotion and memory, which is the reason for such a strong association between certain smells and passion. Olfactory receptors can link to other sensory experiences, so stimulating one pathway may stimulate others. Olfactory epithelium is mainly in the nasal cavity. Sensory neurons have a single dendrite as well as a single axon which extends all the way to the olfactory bulb. Neurons in the olfactory epithelium have a very fast turnaround time, which is around 2 months. Chemical signals allow us to communicate and coordinate olfaction and responses. Odorant molecules dissolve in and penetrate mucus prior to binding to olfactory receptors on cilia extending from neurons. A combination of odorant molecules with receptors activates a special G protein called GALF, which increases cellular CAMP, or CAMP. Increasing CAMP concentration opens CAMP-gated cation channels, which causes depolarization. If graded potentials are strong enough or numerous enough, action potentials are triggered. Action potentials travel along sensory neurons, axons, to the olfactory bulb. Population coding is utilized, where the brain uses combinations of smells to create the perception of many different smells. Taste buds are composed of bunches of TRCs, also known as taste receptor cells, as well as support and regenerative basal cells. Taste receptors are on other regions of the oral cavity, including the palate and the cheeks, not just the tongue. To be tasted, a substance must first be dissolved by saliva and mucus. It then interacts with the apical membrane protein on TRCs, causing a signal transduction cascade ending with the release of chemical messenger molecules from a TRC. Chemical signals released from TRCs activate primary sensory neurons and synapse on the medulla. Sensory information passes through the thalamus to the gustatory cortex, where information is processed. These signals initiate responses that activate things like the digestive system. Transduction of taste utilizes both channels and receptors. The sensation of sweet, bitter, and umami are associated with GPCRs. Salty and sour sensations are more associated with mediated by ion channels. This is because salty and sour sensations are due to sodium and hydrogen ions, respectively. There are 4 distinct cells involved in transduction: cells 1, 2, 3, and 4. Type 1 are glia-like support cells. Type 2 are receptor cells. Type 3 are TRCs, or presynaptic cells. Type 4 are basal cells, which are precursors of TRCs. Each TRC is tucked into epithelium with a tiny tip protruding into the oral cavity. Tight junctions limit the movement of molecules between cells. The apical membrane of TRCs is microvilli, and its purpose is to increase the surface area of the detecting neuron. Taste is also known as gustation, a combination of 5 basic sensations. The 5 current and accepted universal sensations include sweet, sour, salty, bitter, and umami. Umami is associated with amino acid glutamate and other nucleotides. This is the reason MSG, also known as monosodium glutamate, is used as a food additive in many countries. Each sensation is associated with different physiological processes. For example, sour means there's a presence of hydrogen ions, and salty is the presence of sodium ions. The six tastes. Sweet, bitter, and umami taste sensations utilize type 2 TRCs, which express multiple GPCRs on their apical surfaces. Sweet and umami are associated with T1R receptors. Bitter taste uses 30 variations of T2R receptors. Type 2 cell receptors activate G protein gastupsin, which activates signal transduction pathways. Sour evokes responses from type 3 presynaptic cells. When tasting sour, hydrogen-mediated depolarization results in the release of serotonin, which excites primary sensory neurons. Salt taste cells remain unidentified and unproven as of today. The sixth taste is a non-traditional taste and includes things like fats, menthol, capsaicin, mustard oil, and others, and they utilize non-traditional pathways. Specific hunger is when we lack a particular nutrient and we crave specifically to replace that nutrient. For example, if we are low on sodium or salt in the body, we crave salty foods.